So yeah, as Len mentioned, the project that I want to talk to you about today makes use of large-scale and high-throughput single-cell sequencing to gain some insights into genomes that are from bacterial as well as archaeal phyla that are highly underexplored and which we refer to as microbial dark matter. But before diving into the project itself, I want to just first spend a little bit of time to give you a glimpse into sort of our slightly skewed view of the microbial world and some of the gaps in the branches of the tree of life, which will lead me directly into our project, where we're beginning to fill some of these gaps with sequenced representatives. So I will talk a little bit about what one can do with microbial dark matter genomes, which largely relates to phylogeny as well as phylogenetic anchoring of metagenome reads, since these are reference genomes now, and mining these data for functional novelty. And then lastly, I'll give you a future outlook and just want to provide you a view as to where we are today with regards to covering phylogenetic diversity and give you a daunting, but at the same time, I find extremely exciting view with regards to how much diversity there's still left to be covered and explored. So what you see here is a 16S ribosomal RNA tree, and it's collapsed. It's a bacterial tree, and it's collapsed on the phylum level. And the branches that are in gray are branches of phyla that have cultivated representatives. These also have sequenced representatives, although the sequenced representation is quite biased in that even today, more than 3/4 of all the sequenced genomes fall into only 3 bacterial phyla, which are the proteobacteria, the firmicutes, and the actinobacteria. These red branches that you see, these are bacterial phyla that have no cultivated representatives, and we refer to them as candidate phyla. Now, how many there are, I can't really tell you. In this particular tree, I'm showing probably about 30 of them. But depending on the database you're using, you will get different lists of candidate phyla. You will even get different taxonomies. So what are these candidate phyla? And I just want to show you a few names. The OP divisions, obsidian pool divisions, are, for example, some that you may be familiar with. They were found by Phylogenetic when he sampled the obsidian pool sediment. In fact, he found OP1 through OP12, which are 12 phyla in one sample, 12 new phyla. Spam, spring alpine meadow, may be familiar to you from Colorado. There's a whole range, the Guerrero Negro from a hypersaline mat that was found by Ruth Lay in 2006. And then one candidate phyla that you may be quite familiar with also is marine group A, which is also referred to as SAR 406. It's very abundant in marine environments. So this just gives you a glimpse as to what's out there. Now, do these candidate phyla have sequenced representatives? Some of them, yes, they do. And that is thanks to the great advances in metagenome binding and assembly. And Jill Banfield has spearheaded this effort, and she's been very successful in pulling out genomes out of metagenomes. And you also saw yesterday a talk from Eric, where he nicely showed how he can get quite complete or even complete genomes from hypersaline environments using metagenomics. In addition to that, there have been great advances in single-cell genomics, and various candidate phyla are now slowly popping up in the database that are derived using single-cell sequencing. And Paul, I believe after me, will talk a little bit about that with regards to OP9 candidate division. So these were the ones we were after. Now, what did we do? We took samples from 9 sites that we selected, and we selected them because we were already familiar with them. A lot of these samples were from existing CSP projects, and we already had a rough idea as to what the diversity profile looks like. So we knew that a lot of them already had a high percentage of these candidate phyla, which we were after. So what you see here are these sampling sites. In blue are marine environments. That's the Hot Station in Hawaii, and the Gulf of Maine, tropical gyre in the south right here. We have a freshwater and brackish water sample from the Homestake Mine in Sarkanath Lake in British Columbia. In red, we have 2 sites that have sort of thermal activity, which is the Great Boiling Springs in Nevada, in Great Basin, and then the Eastern Pacific Rise, which is down here. And then we have a sediment sample from Greece, the Etoliko Lagoon, and a reactor sample. And as I mentioned, a lot of these we already knew were of highly underexplored diversity. And just to give you an example, this is a 16S pyrotag data of one of these sites, which is the Sarkinev Lake. And if you look at the number of tags that were assigned to candidate phyla, nearly 50% of these tags are assigned to candidate phyla. So these were exactly the samples we were after. So what did we do? We sorted these 9 samples. We isolated over 9,000 cells. Out of these, we had over 3,000 successful MDAs, which we were able to screen using universal 16S primers to figure out what these cells are. And then we genome sequenced and assembled about 200 of them, which resulted in 200 single-cell draft assemblies. Now, I do want to point out from the day that we did the sequencing to the day where we actually ended up with these final assemblies took us nearly a year. That was in part because we had to figure out, we were still in process to figuring out how to best assemble these single-cell genomes. At the time, there weren't really specific single-cell assemblers around yet, which exist today. So we developed our own pipeline. In the process, we also learned a lot about QC. Contamination is an issue with single-cell genomics. So there was a lot of nucleotide signature analysis, a lot of blast analysis, a lot of phylogenetic tree building to make sure that what in the end we have is really from the single cell and we don't have any issues with contamination. So who are these single-cell genomes? What you see on the right-hand side, and I know this is difficult to read, but I just want to give you a glimpse as to who they are. This is a 16S tree. And actually you see the archaeal classes up here and the bacterial phyla down here, and anything that has a color dot sort of correlates with our sampling keys from the previous slide. And these are the phyla for which and classes for which we have single cells. To make it easier for you, I also have a pie chart, which is easier to look at. So we have some of the OP divisions, quite a few of the obsidian pool divisions, CD12, we have EM9, 19 here, and EM3, some of the GN divisions, and KB19, and so on. So we're covering quite some diversity. Now, you have these single-cell genomes, and I don't want to belabor you with assembly statistics, and I'll move through this quickly, but I do want to give you sort of an idea what to expect if you sequence 200 single-cell genomes. What do you get? Now, we get anything in terms of assembly size from a few hundred KB to 2.5 megabases, generally in about 50-ish plus minus contigs. With the largest contig being about 400 KB. Now, what we can do is, for some single cells, we have sort of replicates where we have the same OTU represented by different single-cell MDA products, and then we can co-assemble them. The way we co-assemble is generally we did an ANI comparison. We looked at average nucleotide identity across these single cells, and if it was greater or equal 97%, we started a co-assembly. Now, so we had 21 OTUs, which we were able to co-assemble, and the co-assembly statistics is what you see here, and as expected, they improve. So these single cells complement each other, and you can start to generate more complete genomes. This is also illustrated in this genome completeness map here. Again, sort of the median for our single cells, we get about 35% of the genome, but really anything from a few percent to up to the whole genome. And as we start co-assembling, we're really pushing the boundary higher in getting more complete genomes. And this also led us to get 7 population genomes through these co-assemblies, where we had an estimated genome completeness of over 90%. So moving on. Now we have these 200 single-cell genomes. What do we do with them? First of all, we did want to build trees because these are genomes that didn't exist before, so we really wanted to build genome trees and also see how they compare to the 16S ribosomal RNA-based trees. So a tree approach that we used was we used different marker gene sets. We had up to 38 marker genes. We used different sets because not all of them are universally distributed. We also used different taxon configurations, so to say different ZAG subsets. And that was simply because not all of the ZAGs had all of the marker genes. So we took, ZAGs here stands for single amplified genomes. So these are our single cell genomes. So we took single cells with 20, 30 marker genes. In addition to that, we used different phylogenetic inference methods. And then we also used different outgroup configurations. And we started building trees, many trees, dozens of trees, many dozens of trees. And if I say we, I really mean Christian Rinke, who did this work, and he's sitting in the audience here. So he built many, many, many trees. And this is him right here, and he had a poster yesterday. He likes trees. So what does this data tell us? So what I'm showing you here is a phylogenetic tree, a genome tree. So based on these exact up to 38 macrogenes before we added our single cell data set. Now this is the tree after we add our single cell data set. So we're really beginning to fill in a lot of these gaps and filling out some of these branches. I know these trees are difficult to see on a big slide, but I do want to zoom in just on a few of these branches, just to highlight a few things that we found in this tree. So one area that you see right here, that's the PVC superphylum, the planktomycetes, Chlamydia, Woyke microbial. And there are two additional phyla that are associated with that. One of it is the candidate phylum OP3, and the other one are the Lentisphera. Now, these were previously proposed to be part of this PVC superphylum, and in this case, we were able to confirm it, and in fact, our OP3 genome had also encoded the PVC superphylum signature gene. So that was a nice confirmation. Now, in addition to that, we also saw some discrepancies, so not everything that we saw was consistent with the 16S tree. And one of these examples is here in the FCB superphylum. So within this superphylum, we were able to associate additional candidate phyla. And these are, for example, WS3, the SAR406. So these form very stable groupings, very stable affiliations, as well as WWE1. Now, previously, it wasn't fully clear where WWE1 belongs to. Originally, it was proposed to be its own phylum. Then more recently, it was proposed to be part of the spirochetes, a class within the spirochetes. And our genome tree now, and the genome data was very limited in the past. We generated a lot of WWE1 genomes, and that really helped us to place it properly. And in fact, it is its own phylum. It's not part of the spirochetes. That's at least what our data strongly suggests. Another branch in the tree of life that's very interesting is a superphylum that we ultimately found or came up with. We call it the Pataski bacteria. And what this is a very stable association between these three candidate phyla. Again, previously there wasn't much sequence representation, but now with our single-cell genomes, we can find this robust affiliation. And that is between the candidate phyla GNO2, OD1, and OP11. And again, we thought that stable association does deserve a name, and that's why we proposed to name it Pataski bacteria. Lastly, and this will be my last zoom in on the tree, this is sort of a zoom into the archaea. We were also able to find a stable association between some of the archaeal phyla, and we give it the acronym DPAN, which is sort of an acronym for the beginning of the phyla that are part of this archaeal superphylum. Okay, so in summary, we provided a first substantial genomic data set for some of the bacterial candidate phyla, such as SAR406, OP3, some of the OP, WS1 and 3, BRC1, EM19 and EM3, and CD12, in addition to NKB19, OXPA106, as well as some of the nanoarchaeota-related archaeal groups. We did resolve various inter- and intra-phylum level relationships, and in addition to that, we did propose new bacterial as well as archaeal superphyla. Now, with these new superphyla, we also are proposing new names for a lot of these phyla. So we do propose names, and I'm just going to give you a snapshot for 18 phyla, where we have quite some genomic representation, and for 2 of the superphyla. Okay, switching gears. Assigning metagenome reads to organisms. So we have all these genomes that didn't exist before in the tree. They should serve as very good references for binning metagenome read data. And I just want to show you a quick example. What you see here is so what we did is we took hundreds of metagenomes, and we tried to bin the reads. And we did the binning without our single cells, and then we did the binning with our single cells. And what you see here is the improved binning when we add our single cells into this data set. And the binning is based on BLASTX, and then we use a Megan assignment using lowest common ancestor. And I just want to point out one example, and that is Sarkanov Lake. So for Sarkanov Lake, before we had our single cells, if we do metagenome read binning, approximately 12% of the reads are assigned to some phylum. So they can be binned, so to say, but the bulk of the data is completely unassigned. Now, if we add our single cells to this database, suddenly we can assign and bin an additional 20% of all the metagenome reads. And what do they bin to? I only want you to focus on these 4 red dots. So the bulk of the reads are over 3% of the reads, for example, binned to OP8, over 3% to WWE1, OD1, and OP9. Now, how can we say that this assignment, this binning, is actually accurate? And so what we did is, or what I'm showing you in the next slide is, and again, I only want you to focus on part of this plot. If we look at, so on the left-hand side, you see the assignment of reads before we added our single cells to the data set. So about 38 of these metagenome reads were assigned to bacteria, so they were assigned to the kingdom level. Now, if we add our single cells to the data set and we redo the binning, these reads ultimately are now assigned to some of our candidate phyla, again, to OD1, OP9, OP8, and WWE1. In addition to that, we want to make sure that these new assignments are actually accurate. They're definitely going to a more detailed taxonomic level, but in addition to that, if we overlay that with the average best blast identity, you see that the reads that are assigned to bacteria here have a fairly low, about 50% blast identity, while if we do the assignment to our now reference genomes, the best blast hit identity becomes significantly higher, which is also in a way a validation that actually previously they were, in that case, I'm not saying incorrectly, previously they were just assigned to bacteria to kingdom level, while now we can more accurately bin them to a phylum. Okay, now switching gears, and now I want to talk a little bit about functional novelty. We have genomes from all these novel groups, and you would expect you want to start mining these data. And we found a lot of interesting features that relate to traits that we found in archaea that were previously only described in bacteria and vice versa. And I'm just going to point out specifically one of them. So sigma factor are very important. They are, so far, we've only known them from bacteria, especially if you look at all the 4 domains. They haven't been, all 4 domains of sigma factors haven't been seen in archaea before. But within our single-cell genomes, we had a few archaeal single cells. Sorry, I'm jumping ahead of myself. So within archaea, you don't have sigma factors. You have the TBP, which is the teta-binding protein. But within a few of our single cells, we in fact do find sigma factors in archaea, so they do exist. At that point, we can't do any experimental work because we haven't been able to culture these organisms yet. But these are quite unique features. Now, in addition to that, we also find stringent response, which has only been described and previously known in bacteria. It's ultimately the response of a bacterium to environmental stress. And that is also a feature that we have found in archaea. Lastly, lytic murine transglycosylase is also something previously not seen in archaea. And we do find that in one group of our archaeal and represented in a few single-cell representatives within this particular taxon. Okay, and then very lastly, we also find archaeal features in bacteria. And in that case, this example is the archaeal type purine synthesis. And very lastly, we also find a horizontal gene transfer from a eukaryote into an archaean, which is also, so far, hasn't been described in the literature, and that's an oxidoreductase. And at that point, I do also want to re-emphasize that we really screened these single cells quite thoroughly. So we're excluding that it's unlikely that this is due to contamination. So very lastly, one more sort of unique feature that we found is a recoding event. And so two of our bacterial single cells that belong to the GNO2 phylum, we found that the opal stop codon was recoded to glycine. And this is something that hadn't been described in the literature previously, although there was very recently, a few days ago, a paper on the SR1 genome where this exact same recoding event was also found. So this is quite highly unique. Okay, moving on to the future outlook. So where are we today and how far do we have to go? What you see here is an updated version of a figure that came out in 2009 in the GIBA paper, the original Genomic Encyclopedia of Bacteria and Archaea paper. And I only want you to focus on these gray lines. So what you see is the phylogenetic diversity, and that is based for a subset of genomes. And it's based on the 16S tree, so it's ultimately the sum of the length of the branches in the tree in the 16S tree. And in dark gray here, you see the phylogenetic diversity for all the cultured isolates. And in light gray, you see the phylogenetic diversity for everything 16S based, so all the environmental sequences are included here. And this is a very moving target. This is as of a couple months ago, and of course this keeps, as we sequence more environments, this keeps growing and growing. But the point I want to make with this plot is that to capture about 50% of the currently known phylogenetic diversity, we'd have to sequence about 20,000 genomes, and these would have to be very well selected as based on their diversity. And with that, I'd like to move into the acknowledgements. There has been a great amount of work going into this project, and I want to thank a lot of the PIs that have been really supportive of this project throughout. Phil Jugenholz, Nikos, Natalia has been indispensable for the metabolism. Fang, Jonathan Eisen, Eddie has always been very supportive of this project. We have a group of PIs that have provided us with all the samples. Ramona Stefanowska, Stephen Hallam, Stefan Sievert, Wenzel Lieu, George Tsiamis, and Brian Hedlund. And then lastly, but most importantly, are the people that have really been driving this work, which is Christian Rinke, who really owned this project and brought it to completion. Patrick Schwientek has been indispensable for a lot of the data analysis, and Alex Serba, who helped us to assemble our first large-scale single cell data set. And there's a few more people that I haven't mentioned on the previous slide. Aaron Darling from UC Davis, Donying Wu, and then again, people that were involved in the sample, Brandon, Esther, Jeremy. Ian Anderson at the JGI has also been extremely helpful for this project. Rex Malmstrom, Stephanie, Susanna, Scott, Janie, and Alex, and Jim, and Lens groups, and of course the IMG team. Without IMG, this project would have been extremely difficult. And last but not least, the Department of Energy and Dan Drall. So, thanks.
Other questions? Seems there's still a lot of work to do.
So that is really, really amazing. You know, I was at Christian's poster last night, and it just blew me away. I just thought about it last night. But really, thinking through your presentation, there's even greater implications, I think, for this approach. You know, I'm thinking that rather than wanting to do metagenomics at all, or even have longer sequences, which would be great too, I'd rather have the single-cell genomes from an environment than any of those, because then you can really put it to that single-cell level. You can potentially get away from different biases in doing it. So it's really a whole new way of approaching things experimentally for me. And I was wondering if you had thought of that, because I'd been mostly thinking about culturing things, getting isolates, doing that. Now I'm like, there's advantages to culturing, but the heck with it, just go.
Definitely. So, I mean, we also, with regards to deciphering population heterogeneity, I would foresee that for metagenome assembly, unless you really have very long reads, that's still going to be challenging. While with a single cell, you know that these came from the single cell, so deciphering populations that are heterogeneous, I think it's going to be easier with single cells. Now, I do have to say, it's not an unbiased approach. It has its own biases. So you're not going to get 100 in, 100 out. But it's still, you can cover quite some ground with it. Yeah.
I think the other thing to point out is also that there's a lot of room for technological improvement. I think she gets on average half of a single genome out, so you're not getting the complete thing. I think is that accurate to say?
Yeah.
So if we can get the whole genome, then you have all the biochemical information there. Right now we only have half, but it's a great start.
Two questions, just quick answers, because I don't want to take too much time on this. But first, have you ever encountered CRISPRs in the sequences you've seen there?
We haven't looked for them. I'm quite certain we would encounter them, but we have not specifically looked for them yet. Yeah.
And the other question was about the laterally transferred gene that you saw.
Yeah.
Can you say anything more about that?
No, it's unfortunately an oxidoreductase, so we don't. We know it's most likely coming from a slime mold, Dictyostelium, but that's about all we know at this point.
Any insights on what makes organisms unculturable?
Not yet, no. A lot of these turned out to be heterotrophs, but we haven't, no. And we haven't attempted. We're working with a few people that are now getting interested in trying to culture some of these particular targets that we had in the study.
So do you have any way to deal with the less abundant species? So I know that you sequence 9,000 cells, and but how can you guarantee that all species or most of the species are sequenced?
So out of the ones, sorry. So the question is, within, if we sort 1,000 cells, how do we know that we would get out 1,000 cells? Was that your question?
No, the question is, well, say in metagenomes, we can always see the reads or contigs from the less abundant species.
Oh,
probably not all of them, but we can see at least part of them. But for single cells, it's not guaranteed that you get all the species in the environment.
Most certainly. So it's a random process. We do the sorting randomly. So by chance, and if you sort enough cells, you could get to the rare tail, so to say, or the rare biosphere. But yeah, you don't know. Again, the process is biased. Only a fraction of the cells lies, and so some of them may be difficult to sort even, or they attach to particles, so we can't even get them into our sort window.
So this is kind of a related question, and maybe the next talk will cover it, but of the 3,300 good amplifications you got 201 genomes. What was the rest?
Not candidate phyla. So proteobacteria, actinobacteria, a lot of different other phyla that we were not after. Yeah.